Hello students, today let us discuss about buffer part 2, fitness of aqueous environment for living organisms. First let us come to introduction. Organisms have effectively adapted to their aqueous environment and have evolved means of exploiting the unusual properties of water. The high specific heat of water, that is, the heat energy required to raise the temperature of 1 gram of water by 1 degree Celsius, is useful to cells and organisms because it allows water to act as a heat buffer, keeping the temperature of an organism relatively constant as the temperature of the surroundings fluctuates and as heat is generated as a byproduct of metabolism. Furthermore, some vertebrates exploit the high heat of vaporization of water by thus losing excess body heat to evaporate sweat. The high degree of internal cohesion of liquid water due to hydrogen bonding is exploited by plants as a means of transporting dissolved nutrients from the roots to the leaves during the process of transpiration. Even the density of ice lower than that of liquid water has important biological consequences in the life cycles of aquatic organisms. Ponds freeze from the top down and the layer of ice at the top insulates the water below from frigid air, preventing the pond and the organisms in it from freezing solid. Now let us discuss water as reactant. Water is not just the solvent in which the chemical reactions of living cells occur. It is very often a direct participant in those reactions. The formation of ATP from ADP and inorganic phosphate is an example of a condensation reaction in which the elements of water are eliminated. The reverse of this reaction cleavage, accompanied by the addition of the element to water, is a hydrolysis reaction. Hydrolysis reactions are also responsible for the enzymatic depolymerization of proteins, carbohydrates, and nucleic acids. Hydrolysis reactions catalyzed by enzymes called hydrolases are almost invariably exergonic by producing two molecules from one that lead to an increase in the randomness of the system. Water and carbon dioxide are the end products of the oxidation of fuels such as glucose. The metabolic water formed by oxidation of foods and stored fats is actually enough to allow some animals in very dry habitats like gerbils, kangaroo rats, camels to survive for extended periods without drinking water. The carbon dioxide produced by glucose oxidation is converted in erythrocytes to the more soluble bicarbonate in a reaction catalyzed by the enzyme carbonic anhydrase. In this reaction, water also functions in proton transfer by forming a network of hydrogen-bonded water molecules through which proton hopping occurs. Green plants and algae use the energy of sunlight to split water in the process of photosynthesis. In this reaction, water serves as the electron donor in an oxidation-reduction sequence that is fundamental to all life. Now let us discuss buffering against pH changes in biological systems. Buffers are aqueous systems that tend to resist changes in pH when small amount of acid or base are added. A buffer system consists of a weak acid, that is the proton donor, and its conjugate base, that is the proton acceptor. Almost every biological process is pH dependent. A small change in pH produces a large change in the rate of the process. This is true not only for the many reactions in which the hydrogen ion is a direct participant, but also for those in which there is no apparent role for hydrogen ions. The enzymes that catalyze cellular reactions and many of the molecules on which they act contain ionizable groups with characteristic pKa values. The protonated amino and carboxyl groups of amino acids and the phosphate groups of nucleotides, for example, function as weak acids. Their ionic state is determined by the pH of the surrounding medium. Ionic interactions are among the forces that stabilize a protein molecule and allow an enzyme to recognize and bind its substrate. Cells and organisms maintain a specific and constant cytosolic pH, usually near pH 7, keeping biomolecules in their optimal ionic state. In multicellular organisms, the pH of extracellular fluids is also tightly regulated. Constancy of pH is achieved primarily by biological buffers, mixture of weak acids and their conjugate bases. The figure shows the pH optima of some enzymes. Weak acids or bases buffer cells and tissues against pH changes. The intracellular and extracellular fluids of multicellular organisms have a characteristics and nearly constant pH. The organism's first line of defense against changes in internal pH is provided by buffer systems. The cytoplasm of most cells contains high concentrations of protein which contain many amino acids with functional groups that are weak acids or weak bases. For example, the side chain of histidine has a pKa of 6. Proteins containing histidine residues therefore buffer effectively near neutral pH, and histidine side chains exist in either the protonated or unprotonated form near neutral pH. Nucleotides such as ATP as well as many low molecular weight metabolites contain ionizable groups that can contribute buffering power to the cytoplasm. Some highly specialized organelles and extracellular compartments have high concentrations of compounds that contribute buffering capacity. Organic acids buffer the vacuoles of plant cells. Ammonia buffers urine. Two especially important biological buffers are the phosphate and bicarbonate systems. The phosphate buffer system acts in the cytoplasm of all cells. The phosphate buffer system is maximally effective at a pH close to its pKa of 6.86 and thus tends to resist pH changes in the range between about 5.9 and 7.9. It is therefore an effective buffer in biological fluids. In mammals, for example, extracellular fluids and most cytoplasmic compartments have a pH in the range of 6.9 to 7.4. Blood plasma is buffered in part by the bicarbonate system consisting of carbonic acid as proton donor and bicarbonate as proton acceptor. This buffer system is more complex than other conjugate acid-base pairs because one of its components, that is carbonic acid, is formed from dissolved carbon dioxide and water in a reversible reaction. Carbon dioxide is a gas under normal conditions and the concentration of dissolved carbon dioxide is a result of equilibration with carbon dioxide of the gas phase. The pH of a bicarbonate buffer system depends on the concentration of carbonic acid and bicarbonate, the proton donor and acceptor components. The concentration of carbonic acid in turn depends on the concentration of dissolved carbon dioxide, which in turn depends on the concentration of carbon dioxide in the gas phase or the partial pressure of carbon dioxide denoted as PCO2. Thus, The pH of a bicarbonate buffer exposed to a gas phase is ultimately determined by the concentration of bicarbonate in the aqueous phase and the partial pressure of carbon dioxide in the gas phase. The bicarbonate buffer system is an effective physiological buffer near pH 7.4 because the carbonic acid or blood plasma is in equilibrium with the last reserve capacity of carbon dioxide in the air space of the lungs. When hydrogen ion from the lactic acid produced in muscle tissue during vigorous exercise, for example, is added to blood as it passes through the tissues towards a new equilibrium in which carbonic acid is increased. This in turn increases the dissolved carbon dioxide in the blood and thus increases the partial pressure of carbon dioxide in the air space of the lungs. The extra carbon dioxide is exhaled. Conversely, when the pH of blood is raised by the ammonia produced during protein catabolism, for example, the opposite event occurs. Concentration of hydrogen ion or blood plasma is lowered, causing more carbonic acid to dissociate into hydrogen ion and bicarbonate, and thus more carbon dioxide from the lungs to dissolve in blood plasma. The rate of respiration or breathing, that is, the rate of inhaling and exhaling, can quickly adjust this equilibria to keep the blood pH nearly constant. The rate of respiration is controlled by the brain stem, where detection of an increased blood partial pressure of carbon dioxide or decreased blood pH triggers deeper and more frequent breathing. At the pH of blood plasma, that is 7.4, very little carbonic acid is present in comparison with bicarbonate, and the addition of a small amount of base, that is ammonia or hydroxyl group, would titrate this carbonic acid, exhausting the buffering capacity. Now let us discuss about how untreated diabetes produces life-threatening acidosis. Human blood plasma normally has a pH between 7.35 and 7.45, and many of the enzymes that function in the blood have evolved to have maximal activity in that pH range. Enzymes typically show maximal catalytic activity at a characteristic pH called the pH optimum. Thus, a small change in pH can make a large difference in the rate of some crucial enzyme catalyzed reactions. Biological control of the pH of cells and body fluids is therefore of central importance in all aspects of metabolism and cellular activities, and changes in blood pH have marked physiological consequences. In individuals with untreated diabetes, that is, the lack of insulin or insensitivity to insulin disrupts the uptake of glucose from blood into the tissues and forces the tissues to use stored fatty acids as their primary fuel. This dependence on fatty acids results in the accumulation of high concentrations of 2-carboxylic acids, that is beta-hydroxybutyric acid and acetoacetic acid. Dissociation of these acids lowers the pH of blood plasma to less than 7.35, causing acidosis. Severe acidosis leads to headache, drowsiness, nausea, vomiting, and diarrhea, followed by stupor, coma, and convulsions, presumably because at lower pH, some enzymes do not function optimally. When a patient is found to have high blood glucose, low plasma pH, and high level of beta-hydroxybutyric acid and acetoacetic acid in blood and urine, diabetes mellitus is the likely diagnosis. Carbonic acid acidosis is treated by dealing with the underlying condition, that is insulin for people with diabetes. Severe acidosis can be reversed by administering bicarbonate solution intravenously. So to conclude, most fundamental to all living organisms is the fact that many physical and biological properties of cell macromolecules, particularly the proteins and nucleic acids, derive from their interactions with water molecules of the surrounding medium. The influence of water on the course of biological evolution has been profound and determinative. If life forms have evolved elsewhere in the universe, they are unlikely to resemble those of Earth unless their extraterrestrial origin is also a place in which plentiful liquid water is available as solvent.